Okay, we're good to go, Detlef.
Good morning. Welcome to today's open research webinar hosted by eLife. This series aims to give early career researchers an online platform to continue to share their research as an alternative to in-person gatherings. I'm sure that many of you, just like myself, is now watching from home. My name is Detlef Weigel. I'm a deputy editor at eLife. I've been with eLife from the very beginning for almost 10 years now. I'm a plant geneticist and evolutionary biologist for the past 20 years, but I actually started out as a neurobiologist as an undergraduate in the 80s. So I'm really excited about the talks today that we have three fantastic talks. You'll see the speakers here. We'll hear from Dr. Miriam Matamales, who is a postdoc at the University of New South Wales in Sydney. She'll be speaking to us about striatal function and its role in goal-directed action, a learning update. Next, we'll have Debanjan Dasgupta from the Crick in London. He'll be speaking to us about perception and encoding of temporally fluctuating odour stimulus in mice. And finally, Alexandra Tzilivaki, PhD student at the Charité in Berlin, Germany, will speak about Interneurons intelligent operation. It's a matter of non-linear dendrites. Each of the talks is going to be 10 minutes. And after the talks, we'll have 5 minutes for questions for the speakers, and then we'll move on to the next talk. To ask a question, you can type it into the chat on Zoom. Make sure you figure out where the chat box is on Zoom, or directly into the Google document. We're joined today by Miranda, Anya, and Naomi from eLife, who are working in the background to support all of us. They'll help me line up your questions. I'll read your questions out loud and include your name where possible. The Open Notes document is also a place for you to contribute shared public notes. We welcome you to do so and to list yourself as a contributor in the list above the speakers for today's webinar. Thank you very much for doing this. Finally, if we just stay on that slide, no, the previous slide. So I'd just like to remind everybody that we are recording the webinar, also live streaming this on YouTube, and ask you please to be respectful, honest, inclusive, accommodating, appreciative, open to learning from everyone else. Do not attack, demean, disrupt, harass, or threaten others, or encourage such behavior. And very important, if you feel uncomfortable or unwelcome on any of these webinars, please contact us by email to events@elifesciences.org. Miranda Nye is watching that email address. And finally, we reserve, of course, the right to ask anyone to leave and or to deny access to subsequent webinar on Zoom. If you need help, chat message on Zoom to the host, Naomi, or directly to Miranda, Naomi, or Anya. All right, so without any further ado, really excited that we are hearing now from Miriam, postdoc in Sydney, on striatal function and its role in goal-directed action, a learning update. Over to you, Miriam.
All right, can you all see me? See my screen? Yep. All right, thank you, Detlef, for your introduction. Hello, everyone. Thanks for joining. Today, I'm going to talk about our new view of striatal function and its role in goal-directed action. Please send some questions, and I'll be happy to answer them at the end of the talk. All right, so we know that the capacity to learn and adjust behaviors that secure resources is one of the primary functions of the brain. And this idea of learning from interaction with our own environment underlines a lot of learning theories that are currently out. One of the most influential one is this one from Rescorla and Wagner that postulates that learning is driven by the discrepancy between what was expected from our actions or from a stimulus and what actually happened. That is when errors in prediction occur. So a major question in both in behavioral neuroscience and computational neuroscience is how is this prediction error implemented during learning? And experiments from Scholz and colleagues provided some insight into the neuronal basis of this process. In their experiments, classic experiments, they first show that dopamine neurons, shown here as all these spikes, individual neurons, they were activated following the delivery of an unexpected reward, which was thought to be signaling a positive reward prediction error. Later, when these monkeys were trained with a pair CS, a tone or a light that preceded the delivery of the reward, dopamine neurons did no longer activate it in response to reward, but then they did activate it in response to the anticipatory stimulus, suggesting that dopamine neurons convey reward prediction error signal and not just signal of the reward. Later, they showed that in those trials in which there was an omission of the delivery of the reward following the pair CS, dopaminergic neurons then drop, the activation of dopaminergic neurons drop below baseline, indicating a negative prediction error. So all these and other evidences indicated that this phasic firing from midbrain dopaminergic neurons conveys a reward prediction error signal in the brain. So the question is now, where is this signal going on? So as a lot of people know, the striatum or CPU is the main target of dopamine transmission in the brain. And to illustrate that, I'm showing you here the reconstruction of the axonal arborization of a single dopamine neuron from the SN. And as you can see, this neuron is projecting all of the terminals to the striatum, occupying a large area of this structure. So if you can imagine, if we want to understand how this dopamine signal is encoded in the brain, we have to have a spatial view or a large scale view of what's going on in the striatum. So the striatum is composed by these spiny projection neurons. As you can see here, they are spiny because they are receiving massive input from cortical and thalamic areas, and they are the larger population in the striatum. As you can see here, they outnumber any other kind of neurons in the striatum. Here in the SPNs in white, and here other kind of other interneurons in red. And interestingly, this population can be subclassified into two equally large subpopulations based on the expression of two types of dopamine receptors. So you have D1 type and D2 type, and then you have like D1 SPNs or D2 SPNs. And as you can see, these two types of neurons are not organized in a topographical manner, but they are just like randomly distributed in a intermingled one with each other and filling all this tissue within the striatum. So because they express different types of dopamine receptors, they respond differently to changes in dopamine levels. And some recent studies are starting to elucidate how these two populations encode intracellularly or at the postsynaptic level this error signal. So one example of these studies is this one from Sabatini's lab, in which they show that by measuring the activation or the activation of cyclic AMP signaling within D1 or D2 neurons, they show that intracellular signal was amplified in D1 SPNs following a positive reward mostly, but in the case of D2 SPNs, it was the cyclic AMP activation occur exclusively after a negative reward prediction error trial. So In the experiment, in the study that I'm going to present today, we set up to study or to answer the question, how do D1 and D2 SPNs encode this dopamine signal across large areas of the striatum in order to adjust learned behavior? And to do that, I set up a wide field high resolution mapping of transcriptionally active SPNs. And for that, I used a marker of phosphorylated histones that are known to be induced by or to be phosphorylated in response to cyclic AMP and glutamatergic signal. And then I use D2GFP animals that allow me to classify these D1 and D2 neurons based on GFP content. So I use a spinning disc confocal microscopy that allow me to have high resolution, a single cell resolution of large areas of the stratum. So as you can see here, I just use like very basic automated methods to detect the transcriptionally activated neurons and then classify them based on GFP into D1 and D2. Then using this data, I could reconstruct where in the stratum that this activation was happening. And in I did that in different regions of the stratum. So to study how D1 and D2 encode these different prediction error signals, we train animals in an instrumental learning task in which they have to learn to press a lever in order to obtain a pellet. As you can see here, after 15 days of training, they become experts on this task. So to really understand or to explore how activated and where D1 and D2 neurons are in response to these differences in dopamine levels, we use extinction paradigm that it's a fundamental form of memory updating that prompts the individual to withhold a previously learned action to reduce this action that no longer secure an outcome. And this is a very powerful change in behavior. It occurs very rapidly. So as you can see here, after 10 minutes, animals just stop pressing the lever. There's no point of pressing if there's nothing to get out of this pressing. And so we got these animals and then we studied how these two systems were distributed across the stratum and how these two overlapped within the space. And as you can see here in expert animals, you see that activation of D1 and D2 occurs in different regions, and the overlap between these two populations is very low, as you can see here. However, in animals that had undergone extinction, and this, I should have said that this is the overlap of all the animals in this group. They show that both neurons now overlap in the same region, specifically in the dorsomedial stratum, which is known to be important for encoding the association of these action outcome contingencies. So the question is why the stratum needs both neurons to encode this reward prediction error. And we hypothesize that D2 SPNs, may modulate plasticity in neighboring D1 SPNs during extinction learning. And so to show that pharmacologically, we use raclopride that activates D2 neurons here, or GBR that activates D1 neurons. And we hypothesize that in this test group in which raclopride is injected prior to GBR will this will block the activation of D1 by this compound. And this is what we see here. This quantification of D2 and D1. You see that raclopride mostly activates transcriptionally D2 neurons. In the GBR group, it's mostly activated D1 neurons. And in the case of, and you can see that illustrated here, and in the case of the raclopride plus GBR, there was complete blockade or major blockade of the GBR activation of the D2 by the D2 neurons. So we hypothesized that activation of D2 SPNs in these discrete striatal territories could influence dopamine-dependent plasticity on D1 and hence reshape an outdated behavior. But to test that, we then went back to this extinction learning paradigm, and then we depleted D2 neurons from the dorsomedial striatum using a toxicogenetic strategy, and then we train animals during the in the same paradigm. So with extinction learning, 10 minutes of extinction learning, as you can see, the both groups, there was no impairment of the acquisition of these contingencies when D2 neurons were not present in the dorsomedial striatum. But then in the following day, when after the extinction, after the first day of extinction, we could see that extinction learning was not completely encoded in those animals that were that D2 neurons were absent from this dorsomedial striatum. You can see that here. So these and other experiments that I have to show suggested that control of D1 SPN's plasticity by this D2 SPN's dependent mechanism is critical for updating previously acquired goal-directed behaviors. So to finish, I just show that in this striatum we have like this population of neurons that is composed by these twin subpopulations. And as I show you in expert animals and during initial learning, these two neurons are transcriptionally active in different parts of the striatum. When this learning needs to be adjusted or refreshed, these two neurons are then occupying the same territories. And we hypothesize that D2 neurons can eliminate or inhibit this learning in the D1 neurons that is outdated. With that, if you want more details, there's a paper that we just published a couple of months ago on this. So with that, I would like to thank eLife for giving me the opportunity to speak to you today, and also to all the collaborators that made this work possible. Thank you so much.
Great, thank you very much, Miriam. Awesome, very interesting. I'll start with the first question. How can the connectivity of the local striatal network support the D2 to D1 modulation? How does that work?
So in some experiments that I didn't have time to show, we found that, and others that were classically obtained with pair recordings, we show that there is a bias in connectivity between the D2 and D1 neurons. So there's more, like using transsynaptic virus, we could show that D2 neurons are more connected to D1 than D1 to D2. So these and probably other mechanisms, like not so much into the electrophysiology or in the connection level, maybe more into the neuromodulatory level, could probably support this transmodulation.
Let me look at what the next question is here that Naomi pasted in here. So this transmodulation, you told us about negative prediction error, like with extinction learning. Does this also happen in other types of flexible learning?
So yeah, in another set of experiments that I didn't have time to show today, we train animals in reversal learning, in which animals are trained to learn the contingency between an action and an outcome, and then we reverse the action and outcome contingency. So they have to learn change in their previously learned actions, but without a negative prediction error. So prediction error should be the same. And in this case, when depleting D2 neurons from the DMS, we also had the same effect. So animals were unable to refresh their previously learned action.
Maybe I'll ask one last question that's sort of a very high level question. So it's really amazing what you can do with these, engineered animals and live imaging and whatnot. Can one connect this to any type of experiments with humans? Obviously not invasive experiments, but other type of experiments in humans, or is that, is there still too far a disconnect between these two fields?
Well, like pharmacologically, for example, like D2 receptor antagonists are widely used clinically. So it would be, these are available and are used largely. So it will be interesting to see how these compounds affect learning instead of just like motor output, probably.
Cool, got it. All right, awesome. Thank you very much, Miriam. Awesome. Thank you. All right. We move on to Debanjan, who is a postdoc at the Crick in London. Have we switched over to the slide? Yeah, we have. So Debanjan is going to talk to us about perception and encoding of temporally fluctuating odor stimulus in mice. Over to you, Debanjan.
Thank you, Ralph. Let me just share my
screen. And this is going to mean 1 minute, Debanjan. So.
Yeah, sure. Okay, thank you very much. So firstly, thanks for organizing this wonderful thing, even in the midst of this corona wave. And thanks for hosting us, well. So yes, I am Debanjan in Russ's lab, and we are interested in understanding how odor temporal dynamics can be encoded and perceived by mammals, like especially mice. So if we look at odor diffusion in the real world, what we see is not like the typical square pulse, what we usually give in a laboratory setting, but if we put up a PID, a photoemission detector, in any of these plumes or any of these diffusing odors, we get to see odors fluctuating as a fast and rapid concentration plume, something like this. This leads up to a basic question as why does these happen, and if these temporal structures are in general prevalent in odor plumes, are they useful for animals or any of these olfactory driven animals like mice? So to answer that, we took up a pattern or method of asking this question and built some turbulent environment in the lab, where we introduced odors at different configurations, like odors placed at different spots, at like over here around 50 centimeters apart, or as a mixture, and used a fan and an object to create a similar turbulence like outdoors. And what we observed first is that indeed the odor plumes had a rich temporal dynamics. And while when we placed odors as a mixture or as a single source, we observed that the plume that was occurring from both of these odors arrived at a very correlated fashion, while when they were just around 50 centimeters apart, they arrived as an uncorrelated fashion. We went further and systematically studied how separation or odor source separation might be quantified as in terms of correlation. And what we observed was that indeed, when we change the odor source separation distance, we indeed get a monotonic decrease or a change in the correlation structure. As you can see in this graph, we can find that the correlation is almost at a maximum of 1 when we have it as a single source or as a mixture of odors, while it drops down to almost 0 when we are just around 50 centimeters apart. This suggested that indeed maybe correlation might be an important clue to understand odor source separation, which might be useful for any of these olfactory driven animals, for example, the mice. So we went back to the literature and observed that there has been reports, especially in the insect world, where temporal dynamics have been seen to be useful or to be encoded in the olfactory system of these insects. However, in the mammals, it is largely a far-fetched story, where there has been really little evidence that temporal dynamics is being used or encoded by mammals like mice. So we turned up to the question of, can mice understand correlation structures in order cues? So to do that, firstly, we needed to build an olfactory device which can reliably demonstrate or deliver odors at a very precisely controlled timing. For example, something like this at around 50 hertz, where we can reliably see that the odor can be fluctuating at around 50 hertz. So we did that using these high-speed valves, and we observed that indeed the signal fidelity was largely similar for all the frequencies that we had used, even up to 100 hertz, while the amount of odor release was tightly controlled. With this, we created this basic plume structure where we have a correlated odor stimuli where two odors were fluctuating at a perfectly in sync phase, and the anticorrelated stimulus where they were completely out of sync. And we wanted to test whether mice can understand can discriminate between these two stimulus. That's one of the basic questions to understand if mice can actually treat odor temporal dynamics in a fruitful manner. While of course we did ensure that the flow in these two stimulus were constant so that we don't have any other cue for the animal to do the task. So now we actually plug that odor device into this homegrown, fully automated operant conditioning system, what we call as the autonomous, where animals can live happily in there for up to a year, and they have free food in there. They live in an enriched environment downstairs. While to get water, they need to climb upstairs, get into this tunnel, and where it is really strictly controlled with this magnetic door. And over there, they get to be firstly identified using an RFID chip on their neck. And once they are there, they are given a specific set of trial. And the trial is to identify either if the odor task that is coming up as a correlated or an anti-correlated odor stimulus. So what we see is that indeed from our behavioral experiment, like, I mean, this is the schema of what we are doing, where half of the animals were treated, were asked to understand if the stimulus is a correlated odor stimulus, and will get a water reward if they answer it correctly, while the other half does the other way around. What we see from our star animal is that indeed they can understand and discriminate correlated from anti-correlated stimulus, and further they can even do that at a very high frequency, even up to 20 hertz or 30 hertz, which is way beyond the sniff frequency. And when we did different controls, I don't have the time to go through all of them, but maybe one of the important ones is the switch control, where in any of these tasks we use valves to create these odor stimulus. So any clicking sound in them can be a problem of an extra cue. So we try to change the valve cues for any of these control, and understood that still then the animal was still able to discriminate them as a correlated or an anti-correlated stimulus. Now, if you look into different examples, we indeed see that most of the animals could actually understand correlated from anti-correlated stimulus as we increase, as we give them the stimulus, as we increase the number of trials. Further, when we increase the frequency, which increases the difficulty, they tend to have a decreasing performance with that. We'll see the entire population of the cohort of animals. We have already done almost 1 million number of trials, and we see that indeed the entire cohort could perform the task very well with a very high accuracy level of around 80% for the low frequencies, and up to 10 hertz or 20 hertz, they are even still way above chance, suggesting that they can do the task quite well at all these frequencies. Further, what is surprising is that the frequency that they can do up to is around 20 hertz or 30 hertz, which is way beyond the sniff frequency. We even tested what happens like for a normal plume, which is not anti-correlated, but rather uncorrelated. We did that for 10 hertz, and we saw that even the animals could understand the task for that case as well. We tested for a different odor pair, which is acetophenone and cineol, for which also it worked quite perfectly. Now, when we looked into the reaction times, we observed that the best performing animals had the chance of increasing the reaction time as we were increasing the difficulty of the task, suggesting that indeed they were taking a chunk of the stimulus rather than just the first blip or in the task, which suggested that indeed they were not just paying attention to the first square pulse, but it was a single odor or a mixture of odors, but actually taking into account the temporal structure of the plume. So what we get when we actually classify the performance based on the reaction times is that animals who are performing better than the chance, or rather who are actually taking into account the temporal structure of the stimulus, do take a lot of time, around 750 milliseconds, suggesting that temporal structure is indeed being used by these animals for doing the task. From all what we have got so far is that correlation among incident odor plumes is an essential signature of source separation, and mice can indeed discriminate between correlated and anti-correlated odor plumes. However, what we still don't know is that whether the olfactory bulb, yes, please. Yes, Daniel. How far? What we still see.
You have 1 more minute, Debanjan.
Yeah. In the olfactory bulb is that the frequency contents in the odor, is it being used or is it not being used? So we turned our eyes towards the early olfactory system, which is the olfactory bulb, and we tried to see what happens in the tufted and mitral cells. We went in through using a silicone probe to find that in general, the rasters did not look like they had a very distinct difference for the different frequencies that we had given. However, when we put the spike timings through a linear classifier, the classifier could indeed discriminate for the performance between the different spike timings. This motivated us to do a patch clamp experiment to find out whether there is submembrane, subthreshold membrane potential discrimination between the frequencies. We pick up two frequencies, 2 and 20 hertz, and picked up the membrane potential from them and cross-correlated that with the PID. What we observed is that what we calculated is a coupling coefficient between for all the cells that we had picked up, and tried to find how much is the coupling coefficient varying for a single cell for a given frequency. And what we observe is that for both 2 and 20 hertz, we find that cells did indeed have a certain number of cells had a coupling coefficient more than 1, suggesting that they could indeed follow the frequency in the stimulus. Further, when we actually gave the temporal structure, that is the correlated to anti-correlated pattern, however, a large fraction of neurons had followed, could not discriminate between the two stimulus, while there are a handful of neurons which had encoded the stimulus differently, and suggesting a nice difference in their subthreshold membrane potential as well as in their PSTHs. This altogether, if I can conclude today, is that naturally spreading odors have rich dynamics, and chemicals coming from the same source fluctuate in a highly correlated manner, weakly uncorrelated even for closely spaced sources. And mice can detect correlation structure up to bandwidths of greater than 20 hertz. We observe that olfactory bulb neurons can follow the frequencies both at 2 and 20 hertz, and there are certain olfactory bulb neurons that can encode temporal mixtures differently. I would like to thank, of course, eLife and Andreas' lab, and Andreas himself for conceiving the project, especially Tobias and Andrew, who have been equal contributors of the project. Thank you very much. Any questions, please?
Thank you, Debanjan. Very cool. This is really amazing that the mice can do this. So we'll start right away with the question. So Benjamin Stauch is asking, can humans perform the same correlated anti-correlated discrimination task? And what would be the associated percept?
So this is a very cool question, and of course the next step. So we actually don't know what happens for correlated to anti-correlated, but I guess people might have known a recent paper from Rafi Haddad where humans have been tested with temporal structures in general for humans, and it seems that they have the capacity to discriminate temporal structure in odors. However, it might not be a very straightforward stimulus for them. So that's
what was the do you remember what the frequency was that they used?
I think so they did not use a frequency, but they were using different spots at different time points in a specific
okay
so they did not have frequency per se.
Right. Cool. Okay. Question from Vinod: can innately aversive odors for mice be converted into attractive odors when the frequent is correlated with another odor? So classical type of learning.
This is yet another cool question. So again, never tested this, but maybe possible. One, what I guess Vinod is asking for is an odor identity change. This is something like what Paul Shishka has been trying, has actually published some aspects of it, where if you pair another odor in a correlated or anti-correlated fashion, you can kind of change the odor identity to some extent, but again, largely untested.
Maybe I'll ask a very a naive outsider question. So at 50 hertz, that really blows my mind. That's not something that you expected, that they would perform so well above 20 hertz, or was it?
Sure. That's actually the big surprise from what we observed from the behavioral task. So because especially the sniff frequency, what we have observed so far is not beyond 16 hertz, even from freely moving animals, which we have tried using completely wireless sniff measurements. A paper which is coming up recently, and that suspects that there should be some sniff level of computation going on in the mice, and we have done some preliminary tests using glomerular imaging. Tobias in the lab has done that, and we could see that animals could actually discriminate at very small discrimination in the glomerular level, suggesting a subsniff level of computation.
So we have one last question from Naomi. So this autonomous cage is a really amazing contraption. How do the outcomes in this setup compare to when you have separate chambers? Are the mice less stressed in the autonomous?
Sorry?
Are the mice less stressed? I mean, the research, I assume, are less stressed because it works by itself, but are the mice also less stressed?
Yeah, I mean, of course, they are less stressed because there is actually very, very minimal human interaction. One, and two is that largely most of the behavioral tests, when they are not done in these kind of automated fashion, they are done in the daytime, while mice are actually nocturnal. So they are stressed with that as well. And here we can see that the mice are doing a lot of tasks in the nighttime, which reduces a lot of stress.
Nice.
More natural.
All right, great. Thank you, Debanjan. Thank you very much indeed. All right, great, brilliant. So we move on to our third and last speaker today, Alexandra Tzilivaki, who is a PhD student at the Charité in Berlin, and she'll be speaking to us about interneurons, intelligent operations. It's a matter of nonlinear dendrites. Alexandra, over to you.
Okay, thank you very much, Detlef. Can you see my screen? Okay, so thank you very much for the opportunity to present my recent modeling work that took place in Poirazi lab in Greece. So let's start very briefly. Interneurons, beautiful cells that constitute one of the main types in the mammalian central nervous system. Interneurons are GABA inhibitory neurons and can be divided into many subclasses. Each particular interneuron subclass has its own features regarding the morphology and its own personality, let's say in pattern, in terms of molecular and protein markers. Multiple interneuron subclasses have a prominent role in executive function as well as in neurodegeneration. In this particular study, we're mostly interested in the dendritic computation of fast-spiking basket cells that belong to parvalbumin positive interneuron family. Fast-spiking basket cells contain very thin and aspiny dendrites with calcium permeable AMPA receptors, low levels of NMDA, high density of potassium channels, and low density of sodium channels in the dendrites. Okay, but in general, and this comes for all interneuron classes, the previous hypothesis that was that these neurons are simple on-off cells, which means that according to the point neuron doctrine, interneurons were considered as neurons with very, let's say, not very intelligent dendrites, rather than with linear dendrites that simply integrate synaptic inputs and are not able to perform any kind of clever computational functions. However, in contrast, recent experimental evidence suggests that in many brain regions, for example, in the C1 or in the cerebellum, multiple interneuron subclasses contain clever nonlinear dendrites. So that was the goal of our modeling study, to unravel whether a linear point neuron or a more sophisticated abstraction like the two-stage nonlinear artificial neural network that we know that represents pyramidal neurons can successfully capture the synaptic integration profile of fast-spiking basket cell in the C3 and in the middle prefrontal cortex. So towards that goal, we utilize anatomical reconstruction from rodents, from the CA3 and the prefrontal cortex, with the red using the dendritic trees and with the gray the action. The first step to answer our questions is to extensively validate our 3D in silico interneurons as to make sure that our model cells have the same electrophysiological properties, respond to the same way as the real thing, the in vivo or the in vitro fast-spiking interneurons. So trust me, these very good models. So the first question that we set to our models is, what is the way that the dendrites correspond after the activation of a uniformly activated synapses from 1 up to 20 synapses. So according to the point neuron dogma, if the dendrites of fast-spiking basket cells were linear, then the actual EPSP amplitude should be something like the dashed linear line you see here. But you see that this is not our scenario here. Surprisingly enough, we found out that in all of our models and around 600 dendritic branches that we tested, dendrites can be divided into two nonlinear types. The supralinear ones with the green, where the dendrite is able to produce a dendritic spike, and the dendrite can be also sublinear, which means that this dendrite is not able to produce a dendritic spike and has that jump here. So I want to highlight here that each particular cell has a different percentage of supralinear and sublinear dendrites, but the two modes coexist in all 8 cells tested. So the natural question that arises is, why are some dendrites able enough to produce spikes and be supralinear and some others are not? To answer that question, firstly, we focus on the active membrane mechanism of the dendrites. And we found out that if we block sodium spikes in the dendrites, all the supralinear dendrites are not still able to produce dendritic spikes and became sublinear. So supralinear dendrites have dendritic sodium spikes. So the next question that arises is, okay, well, we do not have different conductances in the sodium between supralinear and sublinear. Why some dendrites can exhibit sodium spikes and some other dendrites in the same cell cannot? Maybe morphology has something to say to us. And the answer is yes. we found out that in the prefrontal cortex models and in the hippocampal models, we have statistically significant differences in the length and in the diameter of the dendritic branches that belong to supralinear or to sublinear category. And we then tested the volume of each branch, and we found out that both in the hippocampal and in the cortical fast spiking basket cell models, all supralinear dendrites were thicker, contain larger volume values, and lower input resistance. On the other hand, the sublinear dendrites, in our cases, are thinner, contain low volume values and high input resistance. So dendritic volume can very well classify the two integration modes that I referred earlier. So how causal is this mechanism? Is the morphological mechanism? Yes, it is, because thanks to modeling, we are able to change the morphology of the dendrites. So here is the control, what we have in the anatomical data, and then we can set the mean length and the diameter of supralinear branches to all dendrites, and you see that we can transform almost all dendrites to become supralinear. With the same reasoning, we can set the mean length and the diameter of sublinear branches to all dendrites, and then you see that most of the dendrites become sublinear. So to sum up, we have two types of nonlinear modes, supralinear and sublinear. In the supralinear case, we can have dendritic sodium spike, and this can be achieved throughout specific morphological features, let's say the volume that these branches have. So, okay, we next attempt to see the effect of the nonlinearity in shaping the neuronal firing of these neurons. Towards that goal, we activated using a 50 Hz Poisson spike train, excitatory synapses up to 60 in all of our model cells. You see here two indicative cases, and these synaptic inputs can be allocated with different spatial allocation patterns. The first one with a baby blue is the dispersed one, where we, for example, activated 60 synapses in a dispersed way, which means scattered throughout the whole dendritic tree. In the grouped way, which is actually the clustered way, we activated the same amount of synaptic input, but only within a few dendritic branches. So we see here that fast spiking basket cells have the intelligence to understand not only the amount of the synaptic input that is coming, but also the spatial allocation profile of the synapses. Okay, it was previously proposed that the preference of these interneurons to disperse spatial allocation of synapses is due to a combination of the diameter of the branches as well as of the existence of A-type potassium channels. To test these propositions, we did a simulation where we set all the diameter of the dendrites equal to 2 microns, and we blocked the A-type potassium channels. And as you see here, our model predictions agree, as now we don't have this preference. So, okay, so in that case scenario, it seems that the point neuron dogma, the linear point neuron dogma, maybe is not a good approximation to describe that cell in a deep learning network, for example. And if yes, what is the best way, what is the best mathematical formalism to describe that cells? Towards that goal, we implemented feed-forward artificial neuronal networks that have the same parameters, except from the hidden layers. 1 minute.
You have to speed up a little bit, Alexandra.
Okay. So here you see the linear point neuron that represents the point neuron dogma. And here you see the two-layer modular ANN, where we have inside the supralinear and the sublinear dendrites. And you see here that according to regression analysis that we did for all our cells, it seems that in all cases tested and in all cells and for a large amount of synapses, the two-layer nonlinear artificial neural network is superior to the linear one and can very well capture the spike rate variance that is generated from our biophysical models. Last but not least, let me very briefly tell you that we finally investigated the functional implication of our finding. My colleague, George Kastelakis, created a biophysical plausible and morphologically simplified macrocircuit model that includes nonlinear dendrites in the fast-spiking basket cells. George initially trained the model to encode for a single memory. The properties of the excitatory engrams were assessed by analyzing the activity of excitation of the excitatory neurons during recall 24 hours after the learning event. Our results indicate that bimodal nonlinear dendrites of fast-spiking basket cells led to smaller excitatory engrams, that the neurons in the excitatory engrams fire less, and we have an increased sparsity. And this means that this nonlinearity can serve as a result in the treating nonlinearities in memory linking by encoding two memories separated either by 24 hours. And here the results suggest that the dendritic nonlinearities of fast-spiking basket cells might be helped to avoid memory interference in the neuronal sequence. So this is our conclusion. The work is available for you to read, and I'm very happy that we have new experimental evidence that support our modeling predictions. I would like to close by setting a question that maybe it's time to stop thinking of interneurons as on-off simple damped assistant cells, but maybe it's time to consider them as equal colleagues to excitatory pyramidal counterparts that together enable us to learn and remember. Thank you very much and stay safe.
Thank you, Alexander. Thank you very, very much. Brilliant. Again, really awesome. Really enjoyed it very much. All right, so I'm looking at the first question here. So from somebody who says being quite far outside the field. So thinking about supralinear versus sublinear, what would that translate into in computing and learning when you have an ANN or a neuron that behaves in a supralinear or sublinear fashion?
Okay. In very simple words, supralinear and sublinear in any case are kind of nonlinear functions that, and we know basically from excitatory pyramidal neurons that when a neuron has that kind of nonlinearities in the dendrite, it means that the dendrite can translate the incoming inputs in nonlinear way. It can do math. It can divide, it can reduce or add, etc. And this enables the cell to correspond to the information that is coming from afferent axonal from axons in many different ways. Okay, for example, the generation of spike. The dendritic spike is a way to enhance the coming signal that at the end passes throughout the soma. So it's a way for a neuron to understand the synaptic inputs firstly, and secondly, to answer to that inputs in many different ways. It enables the neuron with increased computational capabilities.
Yeah. Okay, cool. Okay, so that question was from Alice Misrai. So thank you. So one more question. This one from Romain Kaz. A two-layer neural network is a universal approximator. So it will obviously do better job in approximating the behavior. Is there any other argument in favor of a two-layer artificial neural network?
Okay. First of all, before answering, I have to say that I present here what is the best ANN formalism for this particular type of interneurons. As I said in the beginning, interneurons are very diverse. So do not take it for sure that this ANN formalism is the most suitable for all other interneuron subclasses. We need to find out the computation of, for example, somatostatin and other type of interneurons. Okay, so actually, we don't love the two-layer nonlinear ANN. We are not in favor of that and not in favor of the linear one. We have to understand and to find out what is the best way to translate these cells. And because we found out that these cells, their dendrites have dendrites that that speak, let's say, speak in a logistic sigmoid function or in a logarithmic function, it seems that the two-layer ANN can better describe them. Okay? So that's it.
Got it. Great. All right. Thank you, Alexandra. We had one more question from Vinod, but I think maybe we can take that offline because our time is almost up. So thank you, all three panelists. Great presentations. Thank you to all those who have asked questions. I realize myself that actually it's pretty stressful. Just being the moderator has been pretty stressful. So it must have been a lot more stressful for the three of you. I should mention this seminar was presented in collaboration with neuromatch. It's an online unconference for computational neuroscience, which continues in a few hours' times and runs until 10 p.m. Eastern Daylight Time, so 3 a.m. British Standard Time. For the full agenda and instructions on how to join, you can go to neuromatch.io/agenda. I should also mention that as deputy editor of eLife, I'm really super happy we are doing this series. Right from the beginning, when we founded eLife, that was actually something that was really important to us, to be more than just a place for publication, but we want to change research culture. We want to change how people interact, how people communicate. And we especially want to serve early career researchers. And this was all very much in the eLife ethos. This is a whole series. And so the next installment is going to be on April 2nd, day after tomorrow at 5 o'clock British time with Katrin Vogt, Megan Creed, and Ilke Spark. So I think that's about it. Let's give our speakers another round of applause, and then with that, we'll sign off. Thank you all.
Thank you very much for the great initiative. Thank you. Bye-bye.
Bye.
Bye.